Okay, so welcome everybody to BIO320. I'm Travis. I'll be your instructor for this course. And then the way office hours works is they're typically kind of open-ended. It is a chance for you as the students to come with questions you have specifically. We can use it to review topics. We can talk about the course itself. If you have anything that's one-on-one, you want to talk about grades, that's something we'll probably either do through email or we'll set up an individual time, because I don't want to discuss individual grades while we're a whole group here, because the entire class is invited to these sessions. Before we get started, I do want to talk about the course itself, because there are some positives that students really like about how this course is run and some negatives that students have kind of vented their frustration in past semesters. So I just want to make sure we're all on the same page from the very beginning and make sure this course is something that is good for you as the student. So the course itself runs off of using cogbooks. So hopefully you've had a chance to kind of explore that and go through it a little bit, read about what it is. And the short of it, it's a series of readings, videos, and activities that take place of traditional lectures. Now, I will not be lecturing in the traditional sense at all, like using PowerPoints or anything like that. Rather, that's what cogbooks is for the online. And the positives to that is that you can access the materials at any time that's convenient for you. You can kind of go at your own pace, essentially leading up to the exam. The exams are on specific days, so do make sure to check the course calendar and make sure that you're prepared on that day to take the exam. With that being said, just a quick note on the exams, you have 24 hours that you can pick any 3-hour window to complete them. So they're flexible in the sense that you can take it whenever you're ready or whatever time is convenient for you that day. And that also being said means that obviously you'll have to do the materials before that exam if you want to do well on the set exam. So obviously I would recommend starting cogbooks sooner than later. If you wait until the day before, the day of, you're probably not going to do that great because they are not open book exams. This is an upper level course. It has some prerequisites. It has general biology series and the chemistry series. So you will be expected to have some baseline in biology. So maybe not everything from the exams is going to be spelled out in cogbooks. So you're going to have to understand a little bit about biology, and then it builds on that in more of an ecological context for the course. Now, since the course is using cogbooks, some negatives that students in the past get frustrated with is they like lectures and they don't like the replacement of cogbooks. So there's kind of that trade-off. So it really depends on the student. But knowing that ahead of time, you're going to have to be more self-motivated, because there's not going to be regular times that we meet other than office hours to kind of go over materials. So it is a little bit self-taught in that sense, where you go over the materials and then use me as a resource to ask questions and go over them in a little bit more detail, and maybe have some practice questions here and there in office hours. So with that, are there any questions off the bat about the course structure itself that anybody wants to talk about? And if not, that's okay. If anybody that views this later has questions, feel free to send me an email or a message through Canvas, and I can answer your questions that way. But I also will say, because this course is online, doesn't mean that you have to be completely isolated from each other as classmates. You can use each other to form study groups. You can contact each other on the class discussion board. Contact each other through email outside of class, ask each other questions, quiz each other, and kind of study that way that you might if this was more of an in-person class. So don't hesitate to reach out to myself with questions or to reach out to each other. And there's also maybe a few things that I'm going to share my screen and kind of just show the course itself real quick to make sure that we understand how to navigate the course. All right. So can everybody see my ASU page right here? So the course Canvas page, I should say. Okay, thank you. Yeah, so it's pretty general. You have over here on the left are all your tabs for the course. So the home page is the syllabus page. So if you scroll down, it has the syllabus, talks about the course, the content, the design of the course itself in a little more detail here, breaks down the grades by percentages, breaks down the points. So with that being said, we have 22 modules that we'll go through, and each module in Cogbooks is worth 10 points. And simply by going through them, you'll get full credit. So you get 220 points, which is equivalent to 2.2 exams at 100% if you just simply click through the Cogbooks. And again, I guess a benefit I didn't mention is, I kind of alluded to, I think, is that you can start them and stop them at your leisure. So if you get halfway through one, a quarter of the way, 3/4 of the way, and you run out of time for whatever reason, you can exit out and you can go back and pick up right where you left off, and your grade will be scored at 100% once you've finished going through the entirety of that module. And then there's 5 exams for the course that are 100 points each. And that's 500 points out of the 720 total then. And the way the exams are kind of structured is generally they're about half true, false, multiple choice, and about half short answer. If we scroll all the way to the bottom, the important part is the course schedule. So where it has when the exams are, and it has their due dates and their open dates. You'll notice that they open on a certain day at 12 a.m. or midnight, and they close the same day at 11:59 p.m. So you got that 24-hour window to take the exam whenever you want. And then your Cogbooks, all 22 topics, those are all due on the last day of the course. So even say you don't get to finish one of the modules for exam 2, and we get to the stuff for exam, and then you take exam 2, you can go back at the end of the semester at any time and actually just finish going through that, and you'll get full credit for that, being technically, past the exam, but it's not late unless it's past the course date. All right, and then real important is going to be the modules tab, where hopefully you started going through this. And you have to do some basic stuff first. You have to go take the syllabus quiz and the academic integrity agreement in order to open the different modules. But then you'll click on these different modules, and what they do is the first one, you have this topics and learning objectives, which are going to be helpful for studying for the exams. So right here are a series of questions that are based off the materials. Sometimes on the exams, exam questions come occasionally directly off of this list, and sometimes they're modified versions of those lists, or they might flip the question, or they might ask you, to explain why in a little more detail or something like that. But if you're able to answer these questions, it'll show good signs that you should have some good knowledge going into the exams. And the way that I would approach these is I would leave these open or write them down in a note somewhere, and I would answer them as you come across the materials in Cogbooks. They're generally in chronological order, so as you go through them, it'll go through the list to make it a little bit easier. But I wouldn't wait until the very end. This will actually give you something to look for when you're in the materials and help you kind of study as you're going through the different materials. And also with Cogbooks, I would recommend going through it at one time. Is probably not going to be enough to get an A on the exam or do pretty well. You might want to go through it multiple times, especially some of the activities that you'll see coming up in later modules. And then when we open Cogbooks itself, we get into this menu and you go through one by one. And one thing that's important to note here is, where is it? So if you see this here, how well did you understand the video? You can say that, you can click on it and it's a slide bar, and you can rate it at 100% you understood it and move on to the next one, or you could rate it low. I don't know exactly what the cutoff is, but the lower you rate it, the more extra materials that it will give you, like some extra videos in particular, and it's going to be related to the page. So this is, an intro video on biodiversity. If it didn't make much sense, you can say that. I would just rate it either at 100 or 0. You can play around. I don't think there's anything different from low to high, but it just gives you some extra videos or extra activities or extra readings that can further explain that topic. That being said, the exam is going to be based on all the base material on here. It's not going to be directly from here. So like if there's any new definitions in here, it's going to be the definitions that come from the primary portion of the page. And then lastly, one thing is ask your instructor a question. If you ask a question here, it goes directly to my email and I can reply to you and answer any kind of question one-on-one through this type of messaging system here. And this is kind of nice because it's on the page. You don't have to recall it and say, this section, I don't understand this. You can just, I don't understand this about this topic, and you go from there. And then I can reply. And then once you're done, you can switch it to 100%. And then I guess it stays on there, but it'll give you some options. Okay, so that's the general breakdown of the course and kind of walking into Cogbooks. I want to go back to the home page because there's one more tab that I want to show you real quick where it says discussions. You click on that, that takes us to our community forum, and you can click on the community forum, and then this is a general discussion board for the class where people can interact with each other, and you guys can you can study on here, you can answer questions about the course for each other. I'll periodically check in on this and see if anything major is missing information that I didn't get through an email, but this is something that I'm not going to check every single day. So it's more for you as students to use to communicate to each other. Okay, so are there any questions now about the structure of the course or anything like that? Okay, so after that, what I would like to do is kind of leave this open-ended. So if anybody has any specific questions, there's a few topics that I might want to bring up if we have time for it, just to make sure we cover some stuff. But I'll open it up to the group if there's any questions on the materials. And this is, for today, let's focus mainly on sections 1 and 2, and we can get to 3 and 4 in the next office hours, which I'll have on Friday, and I'll make an announcement for about a day and a Zoom link for that. All right, so there's no direct questions. Maybe why don't we go through maybe a couple of the topics together and kind of work through these a little bit. So this first one here is talking about estimating the number of species. So it gives us an equation that S equals S sub observed plus F1 times F1 minus 1 over 2 times F2 plus 1. So what is this? What are we trying to do with this equation? What's the general sense? Okay, so in the chat, so it's estimate the number of species found in an area. Exactly, right? So our S here is going to be the estimate of number of species. The S observed is the number of species that we observed during our surveys, during our traps, while we're out there. What is the F1 and the F2? Yeah, so species that we only found one or two individuals of. Great. Yeah, so F1 is just when we go out there and we only capture one individual for that given species. So say it's turkeys, we only catch one turkey or observe one turkey. And F2 is when we see two of those. So say that's squirrels, we go out and we catch two squirrels, and we see two squirrels in the tree, right? So a carrot. How come there's no F3, F4, F5, or etc.? Why do we only include F1 and F2 in this equation, do we think? And this can be like open discussion, right? So I know people are typing in the chat. That's great. You can also, turn your microphones on if you want and speak. We can talk as a group. You can kind of just interject. But participation is going to be very beneficial for office hours to go through this stuff. So somebody from the chat said ratio. What do you mean by ratio? Possibly getting at low and high densities of those types of species. So in short, what it's looking at is there's only one and only two individual, right? So it's interested in the rare individuals. So if we have a few individuals that we find only one individual for of that species when we're out there surveying or trapping, it means that there might be other rare species and we might have missed them. So we might underestimate the total population if we don't mathematically account for the rare species. So then to do the calculation, right, we're given a table and it has a list of species. There's 12 of them total. And then there's community A, community B, and community C. And those are just different regions or different areas that were surveyed or measured. So this could be like a swamp, a forest. This could be forest 2, forest 1, and forest 3, or what have you not. It's just a different area that was sampled. And when we look at, and we do this for one community at a time. So that's important to distinguish. Like this equation right here, we just estimated for community A first. And it asked for, species observed. Now, when we have species observed, that just means did we or did we not see that organism? So just because they're all on this list doesn't mean that they were observed in every community, right? Community A, there's a value greater than 0 for every species. So that value, species observed, is 12. Community B, there's a value greater than 0 for every species. So again, that would be a value of 12. But in community C, we noticed for fingernail clams, diving beetles, water boatmen, and dragonflies, there's no individuals there. So that species observed would actually be 8, just the 8 individuals that were observed. And then if we go back now, let's focus on community A for this example. So we plug in 12 there for our species observed. And then F1 is the number of species that we only had one individual. And we see that there's 3 of those, right? There's isopods, we only found 1. Diving beetles, we only found 1. And dragonflies, we only found 1. So that's why this value is 3. And this value is also F1 is 3. and then 2 is just a constant that's part of the equation on the denominator, and then F2 is the number of species that have 2 individuals. And then we see that ram's horn snails only had 2, aphipods only had 2, everything else had a different value, so that value then for F2 is to plug the numbers in, do some algebra, and we estimate for this community A that there would be really 13 individuals there, right? We saw 12, but we think we missed one because there's actually a handful of rare species there. And then we would do the same thing for community B and the same thing for community C, except with the numbers obviously would be a little bit different. We noticed that for community C in particular, there's no individuals that have only one individual observed. So the F1 value for C, that's going to be 0. So the top line is going to be 0. And then species observed plus 0 is just going to be 8 then for community C. Because we feel confident that it's 8, the number that we observed. We're not underestimating it because there were no individuals that only had one organism, or one species that had one organism, and there were only 2 that had 2. So there's less rare species in community C, so we're less likely to underestimate it. Are there any questions on how we did that calculation? Anything not clear, the variables or where the numbers came from or anything like that? Okay. All right. So then I kind of wanted to ask some questions about this to make sure we understand what we're looking at. So does anybody want to take a stab at what these figures here for the patterns of abundance are describing? Okay, so what it's looking at is these are relative abundance curves, right? And what these are is these are going to be, these dots are essentially categories of number of species. So we have number of species on the y-axis and their abundance category on the x-axis. And this is their species on the left. This is where there's little to no species. So they're super rare, and species on the far right are where there are a high number of individuals for that given species. So where these individual species are in large abundance. And what the pattern generally sees is that most species are at some type of intermediate value. We might see a few rare species over here on the left. So there's maybe what is this, six or 7 species. These might be like large predators where they're not very common in an area, so their abundance is very low. More in the intermediate might be some like grazers such as deer or something where their abundance, there's many different types, or maybe this is birds. That's a better example because we see lots of different types of birds out there. So there's lots of types of birds that have intermediate value for their abundance. And then maybe over here on the right, we see something, there's not a lot of different types out there, but there's a lot of them. So maybe this is like ants. There's only like 2 or 3 species of ants in an area, but there's millions of ants in that given area. So they're in very high abundance, but there's very few species that are in high abundance, as well as there's very few species that are very low in abundance. So with these graphs here, we can kind of get a few different pieces of information out here. We can get ranked abundance versus relative abundance, but we can also get species richness. Does anybody remember how they said in the text that we can count or estimate species richness from looking at these graphs? So species richness, what is the definition of richness? Let's maybe back up a step. So species richness is just the number of species in a given area that was sampled. So with this, we have two different communities, right? We have community B and we have community A. We have their ranked abundance. So ranked abundance is just, this is just low in abundance and this is high in abundance. So how many of a given species are in an area. And this is their relative abundance. So it's a proportion. And species richness, we just count the dots. So there's 1, 2, 3, 4, 5 in community B. So there's 5 different species. And there's 1, 2, 3, 4, 5 in community A. So again, there's 5. So they have the same richness for community A and community B because they each have 5 species, but they have a different relative abundance, or given their ranked abundance. So we can learn differences in species evenness because of that. So for evenness, we look at the slope of the lines, right? So evenness, all these species have the same relative abundance. So their evenness is high, right? This is a very even community for community B. Community A has a steep line, so the relative abundance is steeper in community A. So we know that the evenness, it's less even in community A than it is in community B. And then over here, this was an example with lizards in 2 different populations, or 2 different areas. So now they call it the eucalyptus plantation and a primary forest. So we look at richness. Are they the same or different, do you think, in these 2 communities? Yeah, they're different, right? And how do we know that? Because last time we counted the dots. So there's 1, 2, 3, 4, 5 types of species in the eucalyptus plantation. And there's what, 20 or so in the primary forest. So the species richness is much higher in the primary forest than it is in the eucalyptus plantation. And we can get that from just counting the dots. What about the evenness? Are the populations pretty even, or are they pretty uneven? Yeah, so somebody said they're uneven, exactly, because we know that there's a slope to them, right? And the rank, this slope indicates that they're uneven. There's more of these types of organisms than there are of these types of organisms. Great. Any questions on these plots? Okay. And then these are more at the end. These are like critical thinking questions that have feedback with them. And you'll notice that in the feedback, not all of this information is necessarily directly in cogbooks. The exams are going to be more directly based on cogbooks, but again, this is having that expectation of some general biology, chemistry courses. So that's why you might not find the answers verbatim, give it in cogbooks. All right. And then in unit 2, there are a couple of things that I kind of wanted to talk about a little bit. So the title says heterogeneity enhances diversity. When we're talking about landscape ecology and heterogeneity, what do we mean? Like, what does heterogeneity in that context mean? Yeah, so someone said different habitat types within an area. Exactly. So like, what are some examples of habitat types? So maybe a forest could be one habitat. Yeah, somebody said plains, deserts, ponds, oceans. Yeah, so just different types of habitats, exactly. But then the title says that enhances diversity. Why, if a given region of land, if it has more heterogeneity, which means more habitat types, why is that going to enhance diversity for that region? Do all organisms need the same habitat? So someone said different species have different requirements that are met by each habitat type. No, that's exactly right. So if we have aquatic organisms, there has to be some type of water in this environment for them to be present. So if there's a pond right next to a field that leads into a forest, and we say that that's all the same region, we can have aquatic organisms in the pond, semi-aquatic organisms coming in and out of the forest and the pond itself. We can have animals that need field in that field area, and then forested animals in the forest that need trees for different types of shelter. So exactly right. They have different, the different species have different requirements. So the more habitat variety we give them, or they have available to them, the more diverse that given area can be. Then it gets into a little bit more of like, it makes a statement here. It says larger areas hold more species. And we have multiple examples here with plots where we have island area increasing from left to right. So these are bigger islands over here. And then the number of species is generally increasing. So why is that? Why do bigger areas maybe have more species? Why are bigger areas more diverse than smaller areas? And this builds off of exactly what we were just talking about. So if we think about it in context of scale, let's say we survey Arizona State campus. That's a certain amount of square kilometers. And then we do a survey and we survey all of Phoenix area, Phoenix, Tempe, the greater Phoenix area. That's a larger area. Are we going to have differences in habitat type from the campus area to Phoenix? And if you can go say the whole state of Arizona, right? We increase in scale with our surveys, we're increasing an area, and that given area is going to increase habitat heterogeneity. So that's why if you have a larger island or a larger area, you have a higher probability of having more types of habitat or having higher habitat heterogeneity. Exactly, from the chat. Bigger environment means more and different habitat types for a variety of species to live in. Yeah, so pretty straightforward. And then this just shows kind of a mathematical way to calculate species area, but they don't even give an example. And one thing I'll note here, and for the equations, if they have worked out examples, like the first equation we did for estimating the number of species, you can probably expect that you'll have to maybe do that equation on the exam. For something like this, where they don't have worked out examples, they just give you the equation and some data, and they don't even give you that example, you will not be responsible for that equation on the exam. You might have to know that equation conceptually, like what does a species area curve tell us, or you might have to interpret a plot using a species area curve, but you will not have to do that type of calculation if they don't work out an example. Just a heads up moving forward. All right, so then it talks about habitats becoming fragmented. And what that means is you have, say this green area is this forest, and from development, logging operations, and so on, or this could even be from fires or natural causes or flooding or drought. So over time, you can see the forested area has become fragmented, right? There's some problems with that is it decreases the total amount of area of that forest, right? It also increases the matrix in between, or like the distance between the different patches. It also changes the structure where it has more edge habitat. So that can be positive for edge species because edge species need that edge habitat. So it's going to change the types and maybe the numbers of species. So you might get more types of edge habitat species as well as increase their total number. So it can kind of change the face of the landscape. And if those edge species are parasites or if those edge species are predators, that also has some big impacts on the populations that were originally there. And generally, what they talk about is fragmentation decreases biodiversity because most of those reasons it's listed are not very good for organisms. We talk about larger areas have more diversity. So if we decrease that area, we're going to decrease that diversity. It kind of has the opposite effect that we were just talking about. And these are just some pretty good experiments where they actually took, a large area and they divided it up into, fragmented spaces and increased the total edge. And they looked at how that changed species over time. And in this example, they talk about the brown-headed cowbird, I believe, where this is a parasitic bird, where it actually doesn't make a nest. It lays its eggs in other species' nests. So it picks a host, lays its egg in there. And then most of the time, the host species can't differentiate between their offspring, or it's possible they can't get rid of the egg. So they raise that offspring as their own. And as you can see, this larger one is the brown-headed cowbird, and this smaller one is the host bird. They tend to develop faster than the host species and develop bigger, so they can actually outcompete the native offspring to that nest. And that's why they're kind of called a parasite bird. And they're real prevalent on edge habitat. So when they change this habitat, it increased their number, and it also increases their effectiveness because they tend to hang out in fields. But if you give them all these kind of core ways to go in between, which could be essentially fields, they don't have to go into this deep forest and seek out the forested birds. Rather, they're much more obvious and they're not as well hidden. So they have higher levels of parasitism in this fragmented habitat. All right, so in this next section gets into kind of the nuts and bolts of the equilibrium theory of biodiversity. And it kind of shows it in a very basic plot right here where we have size of an island, so area, and then the number of bird species, just, from low to high. But then it also breaks it up into, it includes distance. So we have near islands, intermediate distance islands, and far islands. And that's distance to the mainland. What is the general pattern that we're able to see through this plot? They're really kind of 2 points that interact. One person said the closer an island is to the mainland, the higher diversity. Yep, that's definitely correct for one of the points. And then the other point is based on what we already talked about. So it looks like if we look at all the points together, as the area increases, so as the island gets bigger, we have a positive slope of the number of birds. So large islands have more birds, and islands that are near the mainland, so these green ones, are also the highest in the y-axis. So if they're near and large, they're going to have the most number of organisms. If they are small and this reddish orangish color says far, these dots down here are showing that small and far islands are going to have the least amount of species. And why is that? What are some reasons that the near big islands have more organisms than the far and small islands? Think about transportation. How do organisms get from the mainland to the islands? There's two main modes, right? They can either fly there or they can swim there. They can also, stow away and, hitch a ride. Maybe a parasite on a bird or something gets taken there. But the main organisms that have to get there, they have to either fly or swim. So if we're thinking of birds, we're thinking of flight. So if they're real close to the mainland, pretty easy to get there. If they're really far away, that bird has to be able to fly that distance. Not all birds can fly very far. So not all birds are going to be able to get all the way to those far islands. So dispersal ability is going to be one key factor. And also we talked about the size of the island is more heterogeneous, right? So bigger islands are more heterogeneous, so size matters. If we have big islands with more habitat types, we not only have more space for more organisms, but we also have more resources for those organisms. So that's why the large islands are more beneficial for biodiversity than the small islands. All right, so then this is kind of an interesting experiment that I'm not sure they would get permits to do again today. This is with E.O. Wilson, where they essentially went in to these mangrove areas and had different sized islands and different distanced islands, and they went in, put up tents, and they essentially killed everything on those islands to check the diversity, and then they looked at how those islands recovered, and what they found, not surprising, what we just talked about, is it has number of species present on the y-axis and time, so how many species recolonize these islands essentially. We look at the ones that grew to the highest numbers. Those are the near islands, right? And the intermediate is in the middle, and the far islands, over time, they recovered, but they had the fewest number of species. So this is a direct example through experimentation of observing that. Okay, and then this is the equilibrium theory of biodiversity discussion. And one thing I want to point out here is, yes, these lines are extremely smooth compared to the previous plot that we just saw, because the previous plot was real data, so that's why it's kind of messy. And this is talking in theoretical context. We still see these general patterns, positive and negative, but they're not really ever going to probably be this smooth. And this plot kind of gives us a new twist, right? We have an x-axis here that is the number of species, and that goes from left to right. So no species are 1 to infinity essentially, so it increases from left to right. But then it has two y-axes. It has rate of colonization in this orange line, and it has rate of extinction in this blue line. What we see here is the number of species on an island, when it's really low, that has the highest rate of colonization. And as we increase in the number of species on an island, we have this negative trend where the rate of colonization decreases. And why do we see that pattern, do we think? Why does it go from a high rate of colonization to a low rate of colonization? Exactly, right? As colonizing species leave the mainland, there are less species left to colonize, right? Yeah, so when there's no species on the island, colonization rate's really high because many species are coming over. But it eventually can go to 0 if all the species from the mainland are on the island. There's no new species left to colonize it, so that colonization rate tends towards 0, or eventually goes close to or to 0. But we see the opposite in the rate of extinction, right? So that rate of extinction is really low when the number of species on the island is low, but really high when the number of species is really high. So why do we see this positive effect with rate of extinction? Kind of the opposite argument, right? If there's no species on the island, there's nothing there to go extinct, right? But as more and more species are colonized and are present, the probability of extinction events goes up. And for other reasons, the more and more species that are on that island, it also increases competition, it also increases predation. So there's species are likely interacting more, which can also lead to potential rates of colonization. Maybe they're bringing over more disease and there's interactions are spreading that disease. So there's a number of reasons why the rate of colonization goes up as the number of species on the island increases. All right, so then this plot makes it, adds another variable in here. Now it has small and large. And the point here is to show that, and it's again focusing on rate of extinction, a small island has a steeper line. So it has a higher value of extinction compared to a large island. And for the reasons we already talked about, a large island has more space, it has more resources, so the probability of extinction is lower compared to a small island where there's less space and less resources and potentially even more interaction because the animals might be right on top of each other. But one thing we didn't point out, what are these dots on this plot? And we could even back it up and just go right here. What is this dot right here? Yeah, so it's an equilibrium point, exactly. So it's when the rates of extinction and colonization are equal. So there's balance right here, right? It's being colonized as well as extinction, so it's able to maintain a population, and this is the optimal or equilibrium number of species on that given island. And what we see here then is we see the point of equilibrium shifts when we change those different variables. A small island has a lower equilibrium or fewer species on the island compared to a large island, which moves to the right and has a larger equilibrium. And then this idea here is just kind of building on itself. So it's showing now it's focused on the rate of colonization, but it's focused on near and far. And the rate of colonization we can see is lower for those far islands, right? They're harder to get to essentially than it is for those near islands that has a higher rate of colonization. And it gets to zero at a lower or a slower after a higher number of species rather than the far islands. And again, that near island has a higher point of equilibrium than does that far island. And then when we kind of put it all together in this plot, now we have rate of colonization for near and far and rate of extinction for small and large, right? So we have all of these, we have size and distance of that island, and we can get 4 different points of equilibrium depending on which combination we have. So the small far islands have the lowest equilibrium point, so they have the fewest number of species on their island, versus the large near islands have the largest point of equilibrium. So they have the largest number of species potentially on that island. Any questions or comments or concerns about this equilibrium theory? As related to biodiversity? And again, anybody watching, feel free to email about any of the topics that we talk about in this video if you're not able to join us. All right. If not, let's move on to corridors, because there's a few points that I want to make right here. So we have corridors promote biodiversity. So let's start with the definition. What do they mean by a corridor? We've got a picture of something right there. What are we looking at? We even use this word in like human technology, like walk through the corridor, right? Yeah, so a connection between fragments. So yeah, perfect. In this example, we have forest on this side. They built a highway through here, which split the forest into 2 fragments, right? So we have forest on this side, and they built essentially a land bridge for the animals to go back and forth so they don't get run over by traffic. So it's a connection between fragments that is meant to safeguard and protect the moving animals. A lot of animals want to move between this forest, but it's risky on the highway. So that's obviously a huge benefit, keeps them out of harm's way from traffic. But what's a potential negative if, say, this is the only corridor they built for this forest? Why could the corridor actually maybe be bad for organisms using it? So let's think maybe an example. Let's say this is elk and they're using this. Um, they have to all aggregate at one area. And elk have predators, right? So let's say we have bear or wolves, maybe, or mountain lion that hunt the elk. If they know, they learn elk behavior. So if the elk are all crossing right here, maybe the mountain lion is just going to sit and wait right here. They're going to bring the prey right to them. So ideally, when you have corridors, you have multiple ones, so it makes the prey species unpredictable, that they can't just get picked off by predators. Because eventually, if the prey species learn the predator over there, they might just avoid this, or they might just go across the road at different places. So then we get back into the old problem of large ungulates getting hit by traffic, which is obviously bad for all involved. So then this next part is just kind of like a little practice of uh, the different habitat types. So on the left, we have worse design, and on the right, we have better design, right? So, and we got to do a compare and contrast. So this is considering reserve size. If we have a circle that represents the area of the reserve that's this size, what would be a worse design? Yeah, exactly, a smaller one. So this small circle compared to this big one, this would be a worse design because it's smaller. There's less total area and there's more edge. So exactly. And now if we talk about number of reserves, they did this one for us. So number of reserves, it's better to have one continuous big one than it is to have the same area in smaller broken up ones, right? Because again, this increases the uh, edge of the reserve. It also makes it dangerous because they have to leave the reserve essentially to go through this intermediate matrix to go from patch to patch. So that could be separated by roads, could be towns or neighborhoods where we don't want large animals like in Africa, elephants moving from one reserve to another. If they're going to do that going through town, that can be kind of dangerous for them as well as the humans that live in between in that town. All right, so this is reserve proximity, and that just means proximity of the patches. So if this is a good design, what would be a worse design, considering 4 patches? Maybe something that's, so yeah, so spread out 4 patches, exactly, right? So we've got 4 very connected patches, and if we spread them out further, there's a more distance that they have to travel and maybe dangerous habitats or even unsuitable habitats. So say we think of, maybe it's not even like danger interaction with predators. It could just be, say these are ponds, right? And we're concerned with turtles and frogs and salamanders. We want diversity to increase, so we want them to go to all the different ponds. If they're really close together, something on amphibian can maybe make that quick trek. But if they're really spaced out, that trek could kill them because they could essentially just dry out or dehydrate moving between those patches. All right, what about reserve proximity? So again, this is going to be how close they are together. And this worst design is like 4 patches, but they're in a linear line. What would be a better design than this linear pattern? Oval, I would agree that if we say it's this oval, that would be better because it's one continuous environment. But what this question is actually getting at is the proximity. So with proximity, before we saw this, right? It's 4 dots and 4 dots. So if this is 4 linear dots, and we think of it in terms of 4 other dots, what type of arrangement would be better than linear these linear dots? So maybe this pattern, right? Because these are all equidistant apart. So we say this from top left, we have, patch 1, 2, 3, and 4. If I want to move from 1 to 2, it's right there. If I want to move from 1 to 4, it's right there. And from 1 to 3, maybe that's not complete equidistance, but it's still, relatively close. These two are equally apart. But if we're over here and we want to move from patch 1 to 2, okay, they're equidistant apart. But patch 1 to 3, we either got to go through patch 2 to get there. So you have 2 times you have to exit the habitat, or you have to go all the way around, and even worse for patch 4. So it's just harder to get in between, between patches when they're spaced out in a linear fashion than when they are spaced out more in a circular fashion or square fashion. All right. So about E, reserve connectivity. So how about we have 4 dots that are not connected at all? And if we're talking about connectivity, it's pretty straightforward, right? Where we just put those corridors in and we connect them. It's better, exactly, the connected dots. So if we have corridors in between, they're technically, or this is a small portion of the habitat that's still intact. It's better than if they have to exit the habitat entirely to move from patch to patch. All right, what about the last one, reserve shape? We only have an oval and a circle. So it says circle greater than skinny oval, right? Yeah, so we're saying that a circle is better than an oval, essentially. Why is that? I agree, but now the question is why? Maybe a hint is think back to the brown-headed cowbird. So someone said less edge habitat. Yeah, so if you stretch the circle out, you can increase the edge habitat. Say it's the same, right? We just literally took a circle and we stretched it. We didn't break it, we just changed the shape, right? What if the edge is the exact same amount? What could be, why is this circle but still better than the oval? It does have to do with edge, but it's not necessarily the amount of edge. Circle is more impressed. I'm not 100% sure what you mean by impressed. Compressed. Yes, I think that's, yes, getting on track of what I'm getting at. So there's more internal habitat in this one than there is here. So its proximity to edge is lower in this habitat than it is in this habitat. Or right, sorry, I flipped that. The proximity to, so it's further to the edge in this habitat than it is in this habitat. So if we have something like the brown-headed cowbird that lives kind of on this outer edge, and we have birds that live internally in the forest, that cowbird bird is going to be able to spot and get into this internal forest easier than it is in this environment. It has to go further into the internal environment compared to this one. So that's why having a more compressed circular pattern is better to have it than a more stretched out, kind of more oval pattern. Very good. Are there any questions about these or comments or concerns? Okay, I think that covers mostly the main points that I wanted to go over. At this point, I'm going to turn it over to the group, and if there's any questions or anything that anybody wants to talk about, we can go back to different sections and do that. So is there anything there? So do we have to memorize formulas for the exam? The short answer is no, but there's going to be different ways in which we will combat that. The easiest solution that you'll get for any exam is you can use a note card, and I'll send out an email with exact details on this. But if there's for any formulas, you can always use a note card and write the formulas out. For a later exam, you're going to get a little bit more leeway to put more information on a cheat sheet, so to say, and I'll explain that again when we get to that exam. But for exam one, at minimum, you're going to be able to use a note card and write down the formulas. Now, that being said, you can't put written definitions of what everything is. You can put like words, and again, like I said, I will make a detailed instructions and an announcement about this. But no, you do not have to memorize the formulas for the exam. I'd rather have you learn the concepts and how to use them than waste time actually just memorizing them. No problem. Any other questions? And they could be, yeah, questions about the course, the exams, and the materials. This is your time as students to talk to me about whatever you'd like about the course. And if not, that's no problem. This is, we're just getting started. So these first two units were, I feel, a little quicker. So we end early today, I'm okay with that as well. If we go late some other days, that's going to happen probably when we dig into some of the deeper materials. So the question is, what kind of questions are on the exam? So the exam is going to be broken into two parts. The first half about maybe a little bit more than half on some of the exams is going to be a series of multiple choice and true false. And then the second half, or slightly less than half of the exam, is going to be a series of short answer questions. And those are going to range in points and number. So the fewest on an exam usually is like 4, and the most I think I put on one is 7. And they'll range anywhere from 6 to maybe 12 points per question for the short answer. So those are a little bit more in depth, and that'll maybe give you a scenario and have you define these two terms and explain why the difference between them. So compare and contrast kind of type question. It might be something like in a later unit, or it might be for example, if we're on something like this, I could say describe an ideal nature preserve, and you have to hit, it's worth 10 points. So maybe you want to list, more than 1 point. Want to talk about the number of the reserves, the reserve proximity, and the reserve connectivity and the reserve shape. Those would all be ways to get up to those 10 points. It's not a straightforward formula. Just because it's 10 points on the question doesn't mean you have to make 10 sentences or 10 points. But if it's a 6-point question versus a 12-point question, do expect to write a little bit more for that 12-point question. But for all of them, you should be able to answer them in full in anywhere from 4 to 5 sentences. It's going to be more than 1 sentence for the questions as well. So just a kind of a heads up on the exam questions for the short answer part at least. Good questions. For your classmates that can attend will also appreciate this. Any other questions from the group? And if there's nothing else right now, I'll go ahead and say we can call it. If anybody thinks of anything, you obviously feel free to send me an email or a message through Canvas. Ask questions using that feature at the bottom of the Cogbooks pages. As well as we're going to have another office hours on Friday that I'll make an announcement about tonight about the time for it. So we'll have a chance to go over sections T3 and T4 during that time. And we can go back to T1 and T2 towards the end of that session if anybody has questions that they didn't get to it yet, or they didn't think of them until we maybe talked about it today or saw this on this video. But yeah, so I want to thank those that attended and asked questions, and also I will, for those attending, I will post this on the Canvas page within the next hour. It's going to take a little time to download, but it'll be up tonight. So thank you for attending, and welcome to Bio 320.